let's finish out this section. So this is the idea of synteny. So you can see on the left we have the human genome, on the right we have the mouse genome, and the human genome is color-coded so that each piece that corresponds to a piece of the mouse genome is in the same color. So like everywhere you see these nice brilliant blue colors, those actually come from chromosome 15 of the mouse. That's the thing that they match. And so these are called synteny, which is essentially homologous things that appear in different places. So it was the same thing, but it's moved around. Often it will not be a single gene. It'll often be a chunk of DNA, potentially containing multiple genes that'll move. Just because if the chunk has several genes that work together, which often happens, and they move together, then they'd potentially be under the same regulation, like that part of the chromosome would be open or shut. If you had some here, some there, you might need to open up two chromosomes to get at the genes, and that might have a deleterious effect if it was something that you needed to have rapid response. Just, sort of conceptualizing why this might work this way. Okay, so there's all the red parts, and look at it, bam. There. Okay, so the tree of life, as we understand it, this thing up here, is reconstructed there's actually different ways to do it. You can do it from like ribosomal genes, because all cells, bacteria, archaea, and eukaryotes, have ribosomes. Those ribosomes have ribosomal RNAs. The ribosomal RNAs are homologous. They do the same thing, they fold up the same way, they look the same, and they are related. And so that's one way. You can make a better tree, though, if you take in more genes. So if you include more genes or you include proteins, something like that, you can actually have hundreds of genes that are conserved all the way across the tree, at least in some branches of it, and you can build a much, much better version of the tree. And so this is actually a reasonable version up here. So here's basically the same thing, and so we're reconstructing a common ancestor. Bacteria over here, archaea there, and eukaryotes here, like us. There it is. People, right in between yeast and corn. Yeah, that's where we belong. Plants, fungus, and animals. Yeah. And all the other animals, by the way, are on this same little teeny-weeny branch. So from sea slugs all the way up to elephants are all itty-bitty little branches that come off of this. And so if you compare all of that, like that's what we call the kingdom Animalia. That's the kingdom Plantae. That's the kingdom Fungi. And then in the old classification, they just lump all of these things, like all these bacteria, stick them all together, call them bacteria, which are now moved up to a domain, but still, there's problems. Ideally, a well-reconstructed taxonomic system should reflect something like equivalent evolution between taxonomic ranks, whatever that means. But taxonomy is an inconsistent thing. Okay. So yeah, you can compare genome coding regions like proteins, non-coding regions, whatever. Or you can compare the proteins themselves. That's a much more powerful way to do it than looking at DNA. DNA works really well when you have things that are very, very closely related. And DNA does not work at all when you start to get into much more sort of hyper-evolved, fast-replicating, fast-mutating things. Like looking at the nucleotide level is just death when you're trying to deal with something like viruses. You can line them up if they're in the same species, but outside of a species, good luck. Okay, so yeah, what's the process? Yeah, I bet you can figure it out. So find the region, find which parts of genes look like they can encode proteins, make those proteins, or hypothesize what those proteins should be, just by using the genetic code wheel to effectively translate them or simulate the translation. And then you could also come at it from the other side. You can say, well, all the cells that have this particular gene are able to digest methylcellulose. And all the cells that do not have this are not able to digest it. Therefore, it's a methylcellulase, something like that, if such a thing even exists, which I do not know. All right, so yeah, you can. There are different tools for identifying gene coding regions. This is going to be more important in dealing with eukaryotic databases. So like Procrustes, yeah, does a nice job. It's I don't know if you know the myth of Procrustes, but it's a guy that would invite you to sleep at my house. He's got a bed, and he says my bed fits everybody perfectly. And so what he does is you hop in the bed, he'll strap you down, and he'll say, okay, fine. And if you're too short, he'll put you on a stretcher and yank you so you fit the bed. And if you're too long, he'll just cut off the extra and say, look at that, perfect fit every time. So that's a neat name for a bioinformatics piece of software. Yeah, and so there's different methods of doing it by comparing to other genomes, to other genes, or by just translating things and saying, okay, this is all the stuff we could make, and then going from there. All right. And so comparison of genes, we're going to do this probably by BLAST. That's a thing that we've already covered, so that's good. Comparison of protein content. So you can do BLASTs and align the proteins. That's fine. But then proteins tend to be organized in pathways, because remember, proteins are like little machines that do jobs. And most proteins can't do an entire job on their own. Like, think of a polymerase protein. You can say, well, that builds the new copy of the DNA all by itself. Which, yeah, yes and no, because it's going to have factors that attach to it. It's going to need to have all the factors that assemble on the origin of replication, make that holiday junction. It'll need some helicases to go out in front and sort of clear the way, give it nice single-stranded DNA. And then what's it going to do for nucleotides? It needs all of the genes that are necessary to turn whatever you just ate into some A, C, G, and T, pronto. So yeah, no gene can do it all on its own. It's rare when there are true singletons. They tend to occur in pathways, or at least in genetic programs, where you'll need several activities at the same time to deal with a particular situation. One way of organizing those pathways is the KEGG, which I think the K is Kyoto, but it's a beautiful pathway database. It's listed in your toolbox, and it organizes genes into processes, or rather proteins into processes, and gives them a number. And so you can say, if I've got a gene that does this particular function, like if you're a biochemist and you figure this out, you can say, okay, if I'm doing step 2 out of a 4-step process that makes RNA or something, then I should look for genes that do step 1 and 3 and 4. Yeah, that's all there is to it. You can compare non-coding regions, and there's a lot more non-coding DNA than coding, at least in mammalian genomes. Yeah, so that's okay. That's a thing that can be done, and it can be done very successfully. I believe that paternity testing works on these non-coding regions, although you'd have to check that. I would say Google that. Don't just quote it back to anybody like I did. Right, and so the impact of this whole process is that things that kind of looked like they belong on this part of the tree when we were organizing based on shape and color, like kindergartners or preschoolers actually, get moved around sometimes when we find out that, on a genetic level, they're closer to this. Because there is no other level of inheritance, really. There's genetic, and you could say epigenetic, but really that's just control of the genetic. And beyond that, everything else is pretty small potatoes. Yeah. Histones, but those are only important in the sense that they control access to the genetic information. Yeah, methylation, only important in the sense that it silences or accents some part of the genetic makeup. So yeah, really, it's down to genes and the things that genes make. That's where it's at. So every little stick is called a branch. Every place where these branches connect, or rather where a lineage splits, is called a node. So there's a node here. There would also be a node down here. And sometimes these get drawn as dots. Sometimes you'll see numbers on the node, and that is it's like a branch support value. There are a couple ways to do this. We'll talk about those later, but the most common way is to make several versions of the phylogenetic tree, each using some of the data. Like you'll take the odd nucleotides, then you'll make a tree based on just the even nucleotides, then you'll compare the trees and you see where do the branches match or where do the branches differ. And you'll say, okay, these branches matched up 75% of the time, they get a 0.75. These matched up, none of the time, 5%. So we won't even show them because they're not well supported across at least plurality of the trees. So there you go. And then the one that's at the end, which is the original ancestor, is the root. And so a root is just a kind of a branch to nowhere. Really, the last common ancestor is whatever thing, living thing, or things are at this node. So it'd be neat if you could crack it open and see who's in there. But all you get is a distance. So a phylogenetic distance is the distance from the tip to the node. And it's only distance in one direction that matters. We split these out. So these are split out top and bottom just to keep them separate. The only data carrying branches are the ones that are running side to side on here. And a good phylogenetic tree, you will see a scale bar on the bottom, because of course you will. Otherwise, how would you know? And those are usually in substitutions per site, or substitutions per 1,000 sites or something like that. Across the tree. And so that gives you an idea of how much evolutionary distance there is between a human, it's that far, plus a mouse, so it's that far plus that far, etc. Or how much back to the common ancestor of a human and a fly, and it looks like it's about that much. Yeah, see, you get it. And we can convert that evolutionary distance into time. Again, there are several models. We can estimate these things. We can never know for sure that they are precise, but we can estimate them with error bars. We can figure out confidence intervals. We can do statistics that narrow it down to an acceptable range. And then, for example, in some things, we can go out and find fossils that generally corroborate the molecular dates. The two don't always line up perfectly. The two probably never line up perfectly, but they line up well enough that it seems to work. Yeah. Okay, phylogenetic tree estimation. You'll take these unaligned sequences. You can then align them, and so here they are aligned, and look how beautiful they are. So aligning means putting in gaps where you need gaps to make everything that's homologous, that's the same, all the characters stack on top of each other. And so some things like this P and this T seem to be pretty well conserved. So we've got a good idea that those are both homologous. There's only one thing in the middle. It's different pretty much every time, but because the thing's flanking it and because it's part of a big chunk that together the whole chunk is homologous, we can take that as a character, this point in the alignment. So all these points contribute. The only points that don't really contribute to the mathematics of the alignment are points where we have these little minus signs. So those are a space that we've put in to keep the things that align stacked on top of each other. And those are arbitrary. Like at this point, you can't really phylogenetically compare Y to the absence of Y using sort of parsimony methods. And so we exclude points where there is relatively thin data, where, for example, fewer than 50% of the sequences actually have representation at that position. All right. Once you get your phylogenetic tree, you can look at it several ways. So this tree up here can be equivalent to this tree down here, except that this one has a root. If you were to take off that root, this is a different way that you can visualize the same tree. And if you haven't gone to the trouble of rooting the tree, which can be quite difficult, you got to have a good outgroup. You got to make sure the outgroup is truly homologous and an outgroup that is a group which is outside of these things you're studying based on other inference other than the sequences you're using. Because you don't want to infer something from the data and then use that inference on the same data. That's kind of cheating. Yeah. That's a lot of times a better way to show a tree than with a root. What a lot of programs will do is they will do what's called midpoint rooting. They'll just pick the middle of the longest branches and they'll say that's probably the root. And it may be and it may not be, and you'll never know, but that's why these algorithms sometimes get it wrong. So if you actually want a root, you have to root your tree with an outgroup. That's just how it's got to be done. I don't make the rules, I'm just telling you what they are. Tree topology is branching, and the branching process is the evolutionary process that leads there. So these are all equivalent trees. They have the same branch lengths, and they connect up in the same way. So D and C are always close to each other. And here again, it's only this part and this part that matter. And I think some of these are shown at different scales, but other than that, like I think this one's at half the scale of the others. Other than that, they are identical trees. And you could actually root this. You could root it between A and the BCD node. That would be fine. You could root it between C and D, in which case A and B are very distant outgroups. Depending what you've put in that alignment, there are a lot of possibilities. So in general, alignments work better when you don't have just 4 sequences, because you can arrange 4 in only so many ways, most of which are wrong. What you want are a lot of different sequences in the tree. So it's nice if you can get dozens or maybe even hundreds. It's also nice if you can, instead of having just one gene, you could do your phylogeny across an entire genome, or at least all the conserved parts of the genome. Because remember, you can only align homologous things. Aligning things that are not demonstrably related to each other is a fool's business, because those alignments will count toward the mathematics of parsimony. They'll count toward branch lengths, but there won't be any real indication whether they share a common ancestor. And so you'll just be doing stuff without a particularly good reason. And we don't want to do that. That's not very sciencey. Okay, so there are parsimony methods, there are maximum likelihood methods, where you say, all right, based on this, what's the most likely way that these can be grouped, for example? There are Bayesian methods using Bayesian mathematics. This is a neat way to do it and is generally considered a little more robust than a parsimony or a maximum likelihood. Those are both a little bit on the basic side, and Bayesian tends to get it right when those others get it wrong. There are modifications to Bayesian mathematics, little tweaks, and there are other ways to do this. But I don't believe at this time in history there is a consensus that there is this universally better way. So there will be situations where any one of these is more plausible to use than the next. And that's part of the study of phylogenetics and bioinformatics in general, is being able to use the most powerful method that's available and should give a reasonable result given your data set. And it will vary depending on what you're looking at, when, where, how. So why are phylogenetics so important? They let us classify things, and people love putting stuff in intellectual boxes. They tell us which bits are conserved. If everything is able to change over time and some bits don't change, that means that they changed. but when they changed, the organism died and those genes did not get passed on. Conservation is basically a secondary signal of importance. If something's conserved, it is very likely that it is important to the survival of the organism in whose genome it resides. Yeah, we can learn epidemiology, like where do viruses come from? Well, make a family tree. What are the closest branches to it? Probably that's, the nearest thing. How far is the nearest node from your branch? And you can measure it in time and mutations, whatever. And you say, oh, okay, if it's 500 years distant, then it's probably not a virus that evolved from this other one last week, something like that. We can answer questions about evolution and forensic, whatever. Somebody was a CSI fan, I guess. I don't know. So yeah, we can infer little phylogenies. And the species may have a different tree from some of their genes. Like, for example, it looks like Aedes aegypti and Aedes gambii, these two mosquitoes, have both duplicated their myosin gene. And it looks like this may be on the sex chromosome, the female or male version, which is kind of neat. You have lady muscles and man muscles. I kind of like that. Yeah, arthropods are cool. I hope that's the correct interpretation of this. It may be something less interesting, but here's hoping. Yeah. Or you could, there's different ways to visualize it, but the evolutionary history of a gene is not necessarily the same as the history of that entire species. But if you get all the genes together and look at their evolutionary history as a whole, that's the best job that you can do in reconstructing the phylogeny of that actual group of creatures. So yeah, speciation events, gene duplication events, these are reconstructed, but yeah, interesting. And here we go. There's your virus example. So, all right, SARS-CoV-2. Here are the very early sequences from Wuhan all around the market there. And so what are they closest to? Similar to RATG13, which is a virus that was never grown, but was picked up from bat guano and sequenced. I don't think any of these bat viruses up here have ever been actually grown. It's very difficult to get a virus out of poop and get it to actually work, yeah, as it turns out. And you've also got pangolin coronaviruses that are up here, but you can see most of the relatives are bats. And down here you got bat. This is one that's in a bat but jumped to people. And then down here you got bat, and a couple that have jumped to people or pigs, or also pigs or minks. There you go. And so these are all just different coronaviruses, but this is how we figure out. And so from the branch lengths, you can say, okay, how long was it since this particular virus had a common ancestor with this other virus? And for the distance between these SARS-CoV-2s down here and RATG13, if I remember right, it's about 100 years of evolution. So like before we knew what viruses were, these two things last had a common ancestor. So yeah, this is not a recent new thing by any step. Yeah, and we can reconstruct, so here we've reconstructed different species at this level, reconstructed genera, so everything downstream of this little node is going to be called a beta coronavirus genus. Everything downstream of this node is going to be alpha coronavirus genus, and so on. Yeah, human beings, dividing the world and putting it in little boxes. It's what we do, it's what we're good at. It's maybe what we're best at. All right, thanks.